Hello, my name is Nicole Manzullo, and today I will be talking about Ebola virus. Ebola virus disease, otherwise known as EVD, was first discovered in 1976 by Dr. Peter Pio. He received 2 vials of blood from a Belgian doctor in Zaire. He took this blood and put it under a microscope, where he saw gigantic worm-like structures, which he said were gigantic even by viral standards. When he took this blood and injected it into mice, they were dead within a week. When this happened, he decided to send the blood to the CDC, who reported back to him that it was a new hemorrhagic virus. When he heard this, him and his colleagues decided to go to Zaire and visit the village, which was the epicenter of the outbreak. Ebola was formerly known as Ebola hemorrhagic fever because this is the disease that it causes. According to the CDC, there are 4 species of Ebola that cause disease in humans, and the Zaire Ebola virus, or ZEBOV, is the most lethal of them all because the case fatality rate of this is 70 to 90% if left untreated. The CDC also says that survivors of an Ebola virus infection have antibodies, which are proteins made by the immune system that identify and neutralize invading viruses that can be detected in the blood up to 10 years after recovery. This means that a patient infected with Ebola virus have some long-lasting immunity from it. Now I'll talk about the structure of Ebola. As you can see on this image to the left, there are these yellow structures on the virus. These are glycoprotein, or GP, and this is the envelope protein of the virus. These proteins are embedded in the envelope of the virus, which is a phospholipid membrane, and the virus got this phospholipid membrane when it replicated within an animal cell. The glycoprotein will also help with cell recognition, and the virus will attach to the cell and insert its genetic material inside to infect it. Ebola is a negative-sense, single-stranded RNA virus. Now, how does Ebola enter the body to infect it? It does this through a process called endocytosis, which is depicted in the image to the right. The virus molecules will approach the cell, create infoldings, and are then taken into the cell. This is seen in steps 1 through 3. The cell membrane vesicle is then broken down, and the virus's genetic material and proteins are released into the cellular cytoplasm. Some genetic material will be copied in the nucleus, and some will be translated into proteins. Moving on, there have been many outbreaks of Ebola over the years. The first outbreak happened in 1976 in the Democratic Republic of Congo, formerly known as Zaire. The village that was infected was near the Ebola River, which is where Ebola got its name. The second outbreak occurred in South Sudan, about 500 miles away from this first outbreak. Public health officials assumed that these outbreaks were a single event and an infected person had traveled between the 2 locations. But they were wrong. It was later discovered that these 2 outbreaks were caused by 2 genetically distinct viruses, Zaire Ebola virus and Sudan Ebola virus. The more commonly known outbreak of Ebola happened from 2014 to 2016, and this Ebola outbreak happened in West Africa. The CDC notes that it started in a rural setting of southeastern Guinea, spread to urban areas and across borders within weeks, and then became a global epidemic within months. Many symptoms come along with getting the Ebola virus. As you can see in the image to the right, your entire body can be infected when you have Ebola. Symptoms can appear 2 to 21 days after someone has had contact with the virus. The symptoms also progress in a certain way. First, someone will have dry symptoms like fever, aches and pains, and fatigue. Then they will experience wet symptoms like diarrhea and vomiting. Some other symptoms they can experience are severe headache, muscle and joint pain, weakness, sore throat, loss of appetite, gastrointestinal symptoms like abdominal pain, and unexplained hemorrhaging, bleeding, or bruising. A patient may also have hiccups, a skin rash, and red eyes. Now, how are these symptoms treated? One topical control measure that was successful was a topical immunization through the respiratory tract. This prevented a viral hemorrhagic fever infection in a primate mode. Human studies with influenza indicate that this is the most effective way to restrict infection and viral shedding from the respiratory tract. Although Ebola was mentioned in this study, I have not found any other studies or pages that list this as a type of treatment for Ebola. So other treatments could be providing fluids and electrolytes orally or intravenously into the brain. For example, a doctor could have a patient drink Pedialyte to restore their electrolytes. Some medications that support blood pressure, reduce vomiting and diarrhea, and manages fever and pain would also be beneficial. 2 drugs were also approved as treatments for Ebola. First, in October 2020, the first drug approved was imezab, a combination of 3 monoclonal antibodies. Then, in December of 2020, a second drug was approved called Imbanga, a single monoclonal antibody. Monoclonal antibodies are proteins produced in the lab that act like natural antibodies to stop a virus from replicating after it has infected a person. These particular monoclonal antibodies bind to the Ebola virus's glycoprotein, which prevents the virus from entering a person's cells. Now, how does Ebola infect? Replication and shedding of Ebola. After the virus is engulfed in the cell, the vesicle breaks open and the viral RNA is released into the cell. Some of this genetic material is replicated in the nucleus, and some is translated into protein. New copies of genetic material are used for new copies of the virus. After this happens, the virus then goes into the endoplasmic reticulum. You can see this in steps 1, 2, 3, and 6. The vesicle comes off of the endoplasmic reticulum and then fuses to the Golgi apparatus, where the proteins are then modified, and a new virus comes off of the apparatus to go and be exported from the cell. The vesicle fuses with the cell membrane, and the new virus particle can escape to infect other surrounding cells. This process of replicating and shedding, which occurs over and over again, is the animal virus's incubation period. This is what causes the symptoms. And here in step 8, you can see the vesicle being removed from the cell. Now, how can Ebola be controlled? Through the use of vaccine. The ERV-EBO is a vaccine used for the Zaire Ebola virus. It was approved on December 19th of 2019. And the CDC says that the ERV-EBO vaccine is a replication-competent live attenuated recombinant vesicular stomatous virus, or RVSV vaccine manufactured by Merck. I will first talk about the recombinant aspect of this vaccine. A harmless virus is modified by adding genetic material from the harmful virus, which is Ebola in this case. The harmless virus then replicates, but it doesn't cause any harmful symptoms to a human. The body would be stimulated into making antibodies like B cells that would be effective against the harmful virus if it ever became infected with it. I will now talk about the live attenuated aspect of this vaccine. The virus can replicate in a human, but it is weakened, so there are no harmful symptoms. Well, how is it weakened? A scientist will take a lab critter and infect it with the virus. It will wait until the concentration of the virus is high enough in this lab critter, and then it will harvest the virus again and infect other lab critters. This will happen over and over again until the virus is specialized to a new host. Now that it's specialized to a new host, it doesn't cause any disease symptoms in humans, but it will still promote the B cell production. Currently, this vaccine is not commercially marketed in the US, but it is stockpiled in the Strategic National Stockpile. It can be made available through the CDC for pre-exposure vaccination, though. This is useful for workers like EVD responders, biosafety level 4 workers, and healthcare personnel. Finally, how can it be controlled? One option is killing the virus. Soap is a good way to do this. Soap molecules can break the virus membrane, and with it, the proteins used to attach to a cell. These molecules can also wash viruses off the surface of the skin altogether. Hand sanitizer can also be used. Ethanol can permeate into the middle of the membrane, and alcohol concentrations do matter. 90+% would be needed for it to be very effective. Finally, bleach can be used because it denatures proteins. This means it damages proteins so they lose their 3D shape and become no longer functional, and the virus would ultimately get destroyed. Viruses with a membrane, like Ebola, would have their protein receptors attacked by this bleach. Bleach concentration and exposure time also matter. Someone would have to use 1,000 parts per million of sodium hypochlorite and leave it on the surface for at least a minute for it to be effective. There are many ways to treat and prevent Ebola virus nowadays, so hopefully in the future we continue to see the number of Ebola virus cases diminishing. Here are my references for this presentation. Thank you for your time.